All right. Hello, everyone. My name is Dia Koupantsis, and in this short video, I'm going to talk about heterotrimeric G-proteins. So let's start by a quick introduction or definition for G-proteins. They are guanine nucleotide binding proteins that act as molecular switches to turn on uh, intracellular signaling cascades um, in as a response to the activation of G protein coupled receptors through external stimulus. These trimeric proteins, they have three unique subunits, alpha, beta, and gamma, hence the name heterotrimeric um, proteins. So um, they have the ability to regulate and control different processes, such as regulating metabolic enzymes and transporter proteins, such as controlling the transcription, protein mobility, as well as protein and chemical secretion. So as that happens, a response of the G proteins is going to regulate different functions and pathways, such as learning pathways in the brain, as well as memory and in general and overall homeostasis. However, one of the most important aspects of the structure of these proteins that affects the overall signaling pathway is the covalently attached lipid tails of the alpha and gamma subunit, as you can see here, which anchors to the G proteins in the plasma membrane. And one of the reasons as to why this is important is because in the absence of a signal, the alpha subunit binds or has a ADP molecule that will be bound to it, and the G protein will be inactive. Additionally to that, the binding of the alpha subunit with the GDP and beta gamma subunits, it's going to be with the cognate ligand G protein coupled receptor complex, which altogether are going to move along the signaling pathway, as we'll see later on in this video. So a few important things to notice about the structure of the heterotrimeric complex of the transducing alpha, beta, and gamma subunit are that the beta subunit, to start with, has a seven-bladed beta propeller with four beta strands facing towards, I guess, outwards from the center of the structure. And now these outer strands of the seventh blade specifically provides the first beta strand of the first tryptophan aspartate repeat. What that does is that it causes a polarized interaction between the conserved regions of the tryptophan aspartate residues. So another point is that the gamma subunit takes extended conformation without any intra subunit interaction, so it can happen on its own. And the N terminus of the beta and gamma subunit make a parallel coil conformation. While it happens, the N terminal alpha helix of the alpha subunit, more specifically the alpha transducing subunit, interacts with the first beta propeller blade for the beta subunit. And so the side of that beta propeller is going to interact more specifically with the first and second switch of the alpha subunit. And that structural change is going to cause actually the structural change of the first and second switch. However, the most dramatic change happens in the second switch, where we have the dissociation of the beta and gamma subunit upon the arrival and binding of the GTP to the alpha subunit. So in many G proteins coupled receptors, they have a large extracellular ligand binding domain. And what happens is that when an appropriate protein ligand binds to that domain, what we have is that we have the receptor that will undergo a conformational change that is transmitted to the cytosolic region, which from there is going to activate the trimeric GTP binding protein or G protein. And in this case, in actually in some cases, the active G protein is going to associate with the inactive G protein is going to dissociate or associate with the inactive receptor, while in other cases, it's going to bind only after the receptor is activated. However, in either case, the activator receptor is going to induce a transformational change in the alpha subunit, causing it to dissociate from the GDP molecule. Now, once that happens, we have GTP over here, which is abundant in the cytosol, and now that is going to replace the GDP, and that is going to cause a further conformational change in the G protein, activating both the alpha subunit as well as the beta gamma complex. Now, in some cases, the activated alpha subunit dissociates from the activated beta gamma complex. However, in other cases, they stay together, and in both situations, the activated components can now regulate the target proteins in the plasma membrane. For the GTP to bound to the alpha subunit to activate the target proteins, we have to relay on the signal to other components in the signaling cascade. Eventually, what's going to happen is that the alpha subunit will hydrolyze the GTP to GDP, which in turn is going to activate the subunits. This step is often accelerated by the binding of another protein called regulator G protein signaling, or RGS, and the inactivated GDP bound to the alpha subunit will now reform in an active G protein with the beta gamma complex, which is going to turn off downstream events. And as long as the signaling receptor remains stimulated, it can continue the active G protein through more stimulation. However, the receptor will eventually inactivate the event, and that will happen even if there are active ligands bound to it. Um, so what happens is that the receptor kinase will phosphorylate the protein, and the cytosolic portion of the activated receptor, once the receptor has been already phosphorylated, will bind with high affinity to the rest in the protein, which will inactivate the receptor by preventing the interaction of the G proteins. Now, additionally to that, these proteins act as adapters or adapter proteins and recruit the phosphorylated receptors to claranthine-coated pits, which from where the receptors are going to endocytose, and then afterwards they're going to either be degraded in the lysosomes or activate new signaling pathways. And I know that's kind of a lot of information, but that's what happens, the pathway in a nutshell. So a fun fact about regarding G proteins and receptor specialized neurotransmitters, the alpha subunit of the protein is going to activate one of the adenylyl cyclases, which in turn is going to generate cyclic AMP from ATP, and the cyclic AMP in return is going to activate another membrane-associated enzyme. In this case, is going to be protein kinase A, or PKA. And the protein kinase A is then going to phosphorylate a number of various proteins in or nearby the membrane, and that is going to induce potassium channels. So once these channels close, that is actually going to happen when they're phosphorylated, and they're going to leave the membrane more liable to depolarization. So in many epithelial tissues, protein kinase A activates proteins that transport the working membrane proteins of the tissues towards the apex of the cell, where they are going to do their work depending on what they have to do. Now, it's kind of important to note that the effects of the PKA on neurons are still being worked on. So if you're really interested in this topic, I would suggest that you look more into it. I think it's very interesting and very important. Well, at this point, I'd like to thank you for watching the video, and I hope you have a wonderful day.